Welcome to those of you who are just joining us for the NIBA Science of Therapeutics Symposium on Coronavirus Antivirals. My name is Julien Papillon. I'm an Associate Director in Global Discovery Chemistry at NIBA, and I will be your host for today's session on targeting the COVID-19 protease, with able assistance from my colleague Scott Busby, who will be working hard in the background to select some of your questions for the Q&A. So please submit your questions throughout the talk using the little blue button at the bottom of the window. I know Scott is so looking forward to many questions, as am I. So I think it's fair to say that Mpro is considered to be one of the most promising targets in our fight against COVID infection, with potentials for inhibitors to provide a potent weapon against SARS-CoV-2 and variants yet to emerge, but also potentially a long-term solution to concerns about new coronavirus, with pandemic-causing potential jumping from animals to humans. So we had to have a session on targeting Mpro, and today we have three speakers who will describe their work on this target, including a glimpse on our own effort here at NIBA. So our first speaker is, and I thank you so very much for participating in this symposium, is Dr. Neil London. Dr. London completed his PhD in computational structural biology at the Hebrew University in 2012. He then pursued a postdoctoral fellowship with Brian Schoikert at UCSF. In 2015, Dr. London joined the Weizmann Institute of Science, where he is currently the Alan and Lorraine Fisher Career Development Chair in the Department of Chemical and Structural Biology. Dr. London's lab is developing new technologies to discover and functionalize covalently acting compounds and applies them to drug discovery and chemical biology. Dr. London, the floor is yours.
Thanks, Julian, and thanks for the opportunity to present our work in this great conference. So as Julian mentioned, our lab focuses on covalent ligand discovery. Such compounds have many advantages over traditional non-covalent inhibitors, such as increased biochemical efficiency. They're more potent, they have longer duration of action, and they can be very selective if you're targeting a residue that's only found in your target protein and not in close homologs. And indeed, recently, there are several successful drugs that were rationally designed to covalently target their target protein, including from the last couple of weeks, the first covalent inhibitor targeting KRAS G12C was approved. But there are still some challenges in designing or discovering covalent inhibitors, and that's the focus of our lab. And so we developed technologies to address these challenges, and here are only some of them, including covalent virtual screening, covalent PROTACs, which we recently reported, and covalent electrophile screening. And the reason this is relevant to COVID-19 conference is that Mpro, which, as Julian mentioned, is one of the better targets or more promising targets to act against COVID-19, has a very nucleophilic cysteine in its active site, and for that reason, Frank von Delft from Oxford University and the Diamond Light Source contacted me very early in the pandemic. In February 14, the team at Diamond Light Source already had the main protease cloned and produced after they got the plasmid from Chinese collaborators. Within only 1 week, they were able to determine very high resolution structures of the protease, and the crystallization protocol was robust and was sufficient for crystallographic screening of fragments. Me and Frank collaborated in the past on combined covalent screening and crystal screening, and so they sent us the protein and we ran our electrophilic fragment screen against the target and found a particularly high hit rate of about 150 covalent binders, of which we validated more than 40 in dose response experiments. The Diamond Light Source team has a copy of this library, and so they proceeded to soak hits, both covalent as well as traditional fragment libraries, and collected data from more than 1,500 crystals in a heroic effort in 1 day. And this resulted in 80 structures of Mpro with fragments bound, most of them to the active site, within just a month of the beginning of this effort. So here you can see an overall view of Mpro, and you can see most of the fragments bind in the active site, with a few fragments binding in possibly allosteric site, including the dimer interface. I should mention that we released all of the data as soon as the structures were determined, and later on we were able to publish this effort. If we focus here on the active site, you see that we get very nice coverage of the entire target site. If we focus on specific pairs of compounds, you'll see that there are opportunity to merge these fragments and possibly to improve their activity. But as you can understand, 80 fragments in the binding site is quite a big combinatorial effort, and there are many ways you can merge different fragments. And so at that point, after we published all of our data, we brainstorm how can we really progress something into a more potent inhibitor of Mpro, and in collaboration with Postera, an AI driven medicinal chemistry company from San Francisco, we decided to crowdsource medicinal chemistry design. So Postera set up this website where chemists can upload designs based on the data we released, and then we suggested to synthesize the compounds and to test them. And fortunately, this was the peak of the first wave of the pandemic. Many chemists were locked at home and were looking for ways to help. And so when we tweeted this website and a call for action, the response was not late to arrive. And many computational medicinal chemists just stormed at the challenge and proposed hundreds and hundreds of designs. This is just a random selection, which maybe will enable you to see the diversity of designs that we got from this challenge. Overall, today, we received more than 1,700 designs from more than 400 different people. The project to date synthesized 2,700 compounds, of which about 200 are sub-micromolar inhibitors, and more than 150 were tested against SARS-CoV-2 in cells, against the live virus in cells. So what are the factors that allowed us to progress so rapidly this ambitious project in an academic setting, I must say? So a few force multipliers were a very early decision to focus on complete openness in this project. So open science, all the protocols are published, all the data was released in real time, including crystal structures, biochemical data, the designs themselves. So all of the compounds are publicly available, and as a result of that, no IP can be taken for composition of matter, and the fact that this was all done in the open and that it's patent free allowed many people to contribute and provide in-kind contributions to the project, starting with individuals, consultant with background in medicinal chemistry or computational chemistry, and ending in big pharmaceutical companies that contributed either experiments or time of their employees towards this project. So this really helped us move things fast. Another force multiplier is the ability to use Postera's AI algorithm to prioritize compounds. So out of all of the thousands of the designs that we got, we needed to choose which one we'll focus on. Postera has this product manifold that allows to find the best building blocks out of a vendor's collection, and propose a route. And here you can see an example of a route that was suggested and attempted by a CRO but failed, whereas the manifold suggested route was able to achieve the target compound. And in a study of about 400 compounds from this COVID Moonshot project, where we had on the one hand Postera's predicted synthetic complexity, and on the other hand the empirical data on actually how long it took the CRO to synthesize the compound, you can see there's a very good correlation between the two. So we had a good tool to to prioritize and design the synthetic routes for a compound. The next force multiplier is we had access to high throughput crystallography at XChem, the Diamond Light Source. By now, we published publicly 425 structures of MPRO with fragment bound to them. We have 200 more in the pipeline. And while only 121 are deposited to the PDB, since it takes a lot of time to deposit 400 structures to the PDB, all of them are available almost in real time through Fragalysis, which is an interactive browser-based method where you can compare fragments, inspect binding site, and more. And you have here the web address. You can Google it. So the ability to support the design with structures really allowed fast pace for the medicinal chemistry. And lastly, we tried to make as short as possible design, make, test cycle, where the testing took place both here at the Weizmann Institute, where we used a fluorescence-based protease assay to characterize the compound biochemically, and an orthogonal mass spec-based assay at Chris Schofield's lab at Oxford, where we follow the proteolysis of a substrate peptide. And you can see that overall there's very good agreement between these two orthogonal assays. So for now, I just want to focus on one story or one series, our main series, originating from this effort. In the original fragment screen, we found several fragments binding to this histidine next to the catalytic cysteine, all through a pyridine, forming a hydrogen bond. And then a second series of fragment bound in the P1 pocket, all putting an aromatic group in the same pocket. Actually, if you look closely, you can see that some of these fragments actually open up the pocket a little bit more than the apo structure, which is in sort of pinkish hue. And so one of our our crowdsourced designer in the first round of design suggested to combine these two functions and suggested this aminopyridine on the one hand from one fragment series and this chlorophenyl from another covalent fragment that would be able to capture both interactions. We made the compound and determined the crystal structure, and indeed it hit both pockets as the design predicted, and it had a modest but very ligand efficient IC50 of 23 micromolar. I should mention, we also publish intermediate results such as this, and then there were several follow-up design rounds where people were requested to improve compounds. And so some of the suggestion for improvement of this aminopyridine series really helped us progress it. So overall, today we have more than 800 compounds in this series, and many of them are submicromolar. So one suggestion was to extend to the so-called P4 pocket with this beta-lactam, which improved by almost 7-fold the original compound. Another went downwards to the P1 prime pocket with this cyclophenyl, also improved it. But perhaps the most productive change was from a methylpyridine into isoquinoline that gave us a 30-fold improvement in IC50. As you can see from the crystal structures, which we have for all of these compounds, these are not mutually exclusive. So when we combined the beta-lactam with the isoquinoline, we got another about 3-fold improvement. Same when we combined the cyclophenyl with the cyclohexyl with the isoquinoline, we got another 3-fold. And not that it makes sense from a medicinal chemistry perspective, but for completeness, we made the 3-way molecule, and this has a 100 nanomolar IC50, which is gratifying just from a structural perspective. Another way or advantage we had in designing and optimizing these series is access to folding at home, which is, I think, by now the largest supercomputer on Earth, utilizing hundreds of thousands of CPU cycles. And we use this to run free energy perturbation calculations on virtual libraries of proposed analogs. So for here, in this example, you can see a virtual library of analogs, trying to optimize that P4 pocket where the beta-lactam entered. One result from these very, very large simulation, which overall combined are more than tens of milliseconds of molecular dynamics simulation, showed that most of the analogs were actually worse than the parent molecule, but a few were better. And based on these results, we synthesized a few of the top free energy calculation compounds. And while some of them were equipotent, 3 of them showed a twofold improvement in IC50. Another way we tried to optimize that binding pocket was in our lab using nanoscale high throughput chemistry. We started from this isoquinoline parent at 720 nanomolar. Enamine made boronic acid intermediate for us, on which we could run the Chan-Lam reaction in really small scales using acoustic dispensing. And so we made 300 analogs by installing amines instead of the boronic acid, and then tested them biochemically. And out of these, 2 compounds that we resynthesized and purified, the biochemical assays were done on crude reactions. One of them showed twofold improvement to 400 nanomolar, and the other is twofold weaker. But for that one, we got a crystal structure, and gratifyingly, we see an additional hydrogen bond that we can form through this sulfone. But sort of the most productive series did not go into the P4 pocket, but rather another suggestion from the crowd gave us this benzofuran compound, which gave a twofold improvement over the parent. When we enantiopurified the molecule, it was twice as potent as would be predicted and as the crystal structure showed. And this was the first sort of compound that gave real good results in a cellular assay with the live virus. A lot of the cellular assays were done here in Israel with the Israel Institute of Biological Research, which were able to give us almost 1 week turnaround time on live virus assay, which was really another force multiplier for the project to progress very rapidly. And so we tried to optimize this series. In one attempt, we did the methyl walk, which gave these 2 compounds, which are about equipotent, but modifying the upper methyl to a methoxy gave a nice improvement to an 80 nanomolar compound. And here you can see we're already at half a micromolar in the CPE live virus assay. And this compound also showed nice results against, in this case, in a plaque forming assay against the South African variant. So here you see the 2 micromolar dose of either our compound or remdesivir and the South African variant control. And you can see that while the moonshot compound completely eradicates the virus, remdesivir also already shows escape of the virus at this concentration. So these results in terms of potency were very promising, but of course nothing is that easy. And when we profiled the compounds more, we saw that they have metabolic liabilities. So both the parent has very fast clearance, as well as the methoxy compound. And the current focus of the medicinal chemistry team of the project, I should mention the medicinal chemistry team is mostly comprised of volunteers with ex-pharma experience, probably culminating in hundreds of years in pharma together, led by Ed Griffin from MedChemica. And so some of the more advanced compounds that were designed and tested to tackle this metabolic stability show some promise, such as putting this sulfone on the isoquinoline, improved the stability threefold. This is in vivo PK in mice, and more advanced compound will replace the benzofuran with the cyclic amide, gives an either more promising improvement in stability. And so this is still a work in progress. I just want to share a last vector for optimization. So starting from this parent, we also tried replacing the benzofuran with amine derivatives. You see this didn't give marked improvement. However, adding a second chlorine to this compound gave a very potent compound, 150 nanomolar racemic, with about 100 nanomolar EC50 in the live virus. As I showed you before, optimizing the lower compound gives about 200 nanomolar for the racemic compound biochemically, but with improved metabolic stability. And modifying the free amine really gives potent compounds, with the best one being about 50 nanomolar racemic, 25 nanomolar, probably 25 nanomolar for the enantiopure. So I just want to finish with showing you the medicinal chemistry target product profile that was drafted by Ed Griffin, and we check most of the boxes. So protease assay, 25 nanomolar when we set out to get to 50, 100 nanomolar in Vero E6 CPE assay. In plaque assays, we're at about close to our threshold of 200 nanomolars. Our biggest challenge is PK/PD, where we're trying to optimize metabolic stability. I didn't mention, but we tested in collaboration with Takeda, we tested the compounds against other Mpro proteases, and there are some promising broad spectrum compounds, as well as activity against other variants. We can improve the oral availability, although the molecule is really small, and so we have a lot of hope for that, as well as the solubility, and the half-life is about halfway to what we want it to be. Also didn't have a chance to say, but the compounds are clean in a nanosyn protease biochemical assay of 60 human proteases, no hits, as well as in a safety panel by Eurofins, pretty clean, and so show a little bit of CYP450 inhibitions, but nothing too alarming. Our near-term plan are to pursue lead optimization and progress as quick as possible towards IND. And on that regard, I wanted to announce that Drugs for Neglected Disease Initiative, the NDI, have recently formally adopted the moonshot and will help us progress the projects toward this goal. And we hope to get to an IND by early or mid 2022. This was a hugely global project with too many collaborators to acknowledge. I hope you can see that from this map. So I'll suffice in thanking my own lab members, almost all of which contributed to the project. And I thank you for your attention. I'll be happy to take any questions now.
Thank you, Dr. London. Very exciting to see all this great progress. I don't see very many questions coming through. I certainly have a number of questions, so maybe I can get started. Maybe one thing I missed from the profile of the compounds you showed is maybe pan Mpro activity. Can you comment on that? Is that something that you've profiled, and where do you stand on that question?
Yeah, I didn't show the data. But so most of our compounds show almost perfect correlation between SARS-CoV-1 Mpro and SARS-CoV-2 Mpro inhibition. So between the SARS-CoV, they're almost identical, with selectivity for SARS-CoV over, for instance, MERS and some of the non-pathogenic coronaviruses. Whereas some compounds in the series, not the most potent ones, show promising broad spectrum inhibition across several Mpros from other species as well. So we think we have good leads in the future to develop broad spectrum Mpro inhibitors, although the series we're focusing on now will likely be specific to SARS-CoV-2 and SARS-CoV-1.
Okay, that's great. Thank you. Another question I have that I find interesting, that being a lab focusing on covalency, and you end up optimizing non-covalent ligand primarily. Where do you see that going? Do you think you can get to the goal with non-covalent inhibitors, or are you also exploring covalent handles to maybe go a little bit deeper in terms of efficacy and potency.
It's not nice on you to pour salt on open wounds. Yes, I would really have loved to make to take a covalent drug into the clinic, but we think actually going non-covalent, we think we achieved the target potency with non-covalent, and we actually offer sort of a new modality. We're not aware of any non-covalent, non-peptidomimetic compounds targeting Mpro, so we think this is sort of an asset that explores a new way to inhibit Mpro, and might have advantages both in terms of safety, perhaps in terms of biochemical properties, maybe also in resistance mutations cropping up. So certainly it's nice to have somewhat differentiated asset to add to the mix of Mpro inhibitors.
Great, thank you. I think we're almost out of time, but I do want to ask you one last question. The field sort of started with using these lactams as glutamine mimetics with that hydrogen bond donor that could be problematic, and it's fantastic you found these pyridines and isoquinoline that seem to be a solution. Have you found any other glutamine mimetics that could be of use that you just haven't really talked about or disclosed?
Yeah, we screened many isoquinoline bioisosteres, actually mostly to try and overcome the metabolic stability problems. We think our metabolic liabilities lies on the isoquinoline, and we found a few, not too many, maybe 2 or 3. Again, all the data is available, so you can go to Postera's COVID section on the website and look for the compounds. We found a couple of other compounds that can replace the isoquinoline. They're all aromatic, heteroaromatic substituents.
Okay.
Also,
so you're able to substitute
with yeah, a little bit, but these are not as heavily explored. We have much less structure-activity relationship for those.
Okay, thank you. There is one question from Jay. To what extent are you able to access next-generation polymerase inhibitors for preclinical study, imagining that regulatory agencies and perhaps investigators will desire that development path?
So as for polymerase inhibitors, we have access to remdesivir and to the commercially available compounds from Merck, I think, and we'll try to do combination efficacy trial with those. There's no reason to believe that there wouldn't be at least additive effect, since they were going for alternative mechanisms. So that's definitely something in the development plan, to see the effect with at least the approved drugs.
Thank you. One question around the going back to the beginning of the talk and your flowchart assays. You developed a mass spec assay. How sensitive is that compared to other assays, and how low were you able to go in terms of enzyme concentrations? And a second question related. Did you find it to be better at filtering out false positives as well?
So first, for the facts, the mass spec assay, we use about 325 nanomolar enzyme, if I remember correctly. So it's less sensitive than the fluorescence assay. For the fluorescence assay, we were able to go as low as 5 nanomolar. So actually, for the more potent compounds, the fluorescence assay gives better differentiation. I showed you the correlation between the two assays, so it's pretty good, and I think it helped us dodge some bullets, since some compounds seemed really potent in one assay and not the other, and these turned out to be toxic in cells later, so probably working through some promiscuous mechanism. We don't have full explanation as to why one assay would be sensitive and the other not. Maybe it's due to the concentration and the fact that at the higher concentration, at the mass spec assay, the protease might be more prone for dimerization, and at the low concentration of the fluorescence assay, we may be testing against the monomer and not the dimer. So It's one explanation for discrepancies, but we're not 100% sure. But for sure, we see artifacts that are only picked up by one and not the other.
Okay, thank you very much once again for a great talk and some great answers to all
Thank
these
you.
questions. I appreciate it. Thank you.